Today we're going to be talking about a specific form of motor control, and this is exactly what I'm using right now. It's language. It is an interesting, unique form of motor control, somewhat specific to humans, although there are groups of animals that do have various forms of language, or at the very least learn communication, as we will learn in the next lecture. But today we're going to be focusing on language, mostly talking about work done in humans. So what is language? Language is a system by which sounds, symbols, and gestures are used for communication. It uses words combined according to grammatical rules, and it's expressed in a variety of ways that includes gestures, writing, and speech. When we talk, we are vocalizing, we're generating sound waves, but often there's body language that is used as well, which unfortunately doesn't come through very well through a YouTube presentation like this. And those things are actually somewhat hard to suppress, and they can be very characteristic of what the individual is expressing. We also have written forms, obviously, as well, which is uses a lot of the same centers involved with processing language, but clearly is involving very different muscle groups. So this is where we're going to start to see a lot of different brain systems taking over different modalities. So the general process of language is that it comes into the brain through visual and or auditory systems. You can read text as I'm moving my little laser pointer here, but you're also listening to the words that I'm saying. So there's a visual component and an auditory component. Now the motor system is needed in order to produce language. The more fundamental part of what we think of as language, which is just speaking, that is referred to as speech. And we will be talking about the different muscle groups that are involved with that process. And of course, there's also writing, which we're actually not going to discuss in any detail, but we will talk a little bit about reading, reading using your fingers, mainly through braille. And brain processing between sensory and motor systems is the essence of language. So this is one major place where you're going to see the integration of sensory inputs and then motor outputs. And this is where, somewhere in between, is where we're going to find language coming about and being generated. Human speech is made by the vocal tract, and it involves the coordination of over 100 muscles. That includes all the way down from the muscles that control air pressure coming out of the lungs, all the way up through the larynx, which is right here, in this area, and all the muscles involved in the mouth. All these muscles are controlled by the motor cortex. So the human vocal tract includes the vocal apparatus from the larynx down here all the way to the lips. We refer to this as the vocal tract. The structures above the larynx, including the pharynx, which is right here, the soft palate up here, and the nasal cavity in here, they shape and filter the harmonic series that are generated by the vocal folds, which are right here. The vocal folds sit above the trachea, and air is pushed through the vocal folds in order to cause them to vibrate. And so why don't we talk through this process step by step on the next slide. So the very first step in producing speech involves exhaling air from the lungs and as they are forced through the larynx. The first portion is that there's a contraction of muscles to pull the vocal folds next to each other, and air is pushed through that in order to cause them to vibrate. So these sound waves that are produced by the vibrations of the tightened vocal folds. So as air is forced through, and as you can see, there's a portion here where they're contracted, vocal folds are brought close together, air pushes through, and the vocal folds start to vibrate. And that generates a frequency of sound that is going to be the frequency that you speak with. The tighter the vocal folds, the faster the vibration, the higher the pitch. All of us can increase our pitch a little bit. I've got a relatively low pitch that is my normal speech, but I can increase my speech like this. I can speak with a higher vocal fold frequency. Yeah, just like this. Sound a little bit like Mickey Mouse. Stupid. All right, so you can force these things, and so what I'm doing by that, I'm increasing the muscle contraction strength, tightening these vocal folds a little bit more so that they vibrate faster to increase the pitch. So we have the vibrating vocal folds that is now the source of sound. Sounds that are generated at the vocal folds are modified through various stages of the vocal tract. So we can have this generation of various frequencies at the vocal source, but they get filtered through the vocal tract, passing through the pharynx. They obviously go through the structures of the oral and nasal cavities. You can speak somewhat nasally like this, where I'm forcing all of the sound to come out of my nose. Or I can have it come out of my mouth more, and I can be a little bit more operatic. We can shape a lot of just the sound of where it's passing through our overall vocal tract. But obviously, there's a ton of filtering that's happening from the movements that our mouth is making, controlling the tongue, the position of the tongue, the position of our lips, all of this being leading to a filtering of the sounds that we're generating out of the vocal folds to create the sound that actually leaves our mouths, that are what eventually are what were referred to as phonemes. And then this is eventually the stimuli, the sound stimuli that we hear that we call speech. Speech is made up of fundamental basic sounds, and we refer to those units of sound as phonemes. So every single language will have a suite of phonemes. There are many phonemes that are shared between languages, and then there are other phonemes that are unique to those languages. English has 44 phonemes. We have 24 consonant sounds and 20 vowel sounds. Even though we only have 5 vowels, there are many ways of pronouncing those vowels in slightly different formations. Now, some of these consonants are voiced, which means that you're using your vocal folds when you're making those consonant sounds. Examples of this is B or M or D, for instance. So when I'm making that consonant sound, I have got a little sharp sound that precedes the rest of the phoneme, but during the phoneme, I'm actually producing sound through the vocal folds and generating the vibrations there and then shaping it through the vocal tract. There are consonants that do not use the vocal phones. So these are non-voiced consonants, and these are such as ch, f, or p. So I'm not vibrating my vocal folds when I make those consonants. But when I combine that consonant with a vowel, such as pa or po or poo, now I'm vocalizing, but the vocalization part is falling on the vowel sound. All vowel phonemes are voiced. Po, poo, pee, pe. You can see the vowel portion involves using the vocal folds. Now, when we're talking about phonemes, we can obviously speak them, but it would be a great way, it would be awesome if we could just visualize them. We can. That is done using what is called a sonogram. Sono meaning sound, gram meaning drawing. This is an example of a sonogram. So on the sonogram, we have an X and Y axis. So the X axis is time. That's shown here in this sonogram, time in seconds. That's what the S is here. And you can see the scale here is from 0 to 2.9 seconds. So this is a sound clip that was only 2.9 seconds in length. There is frequency on the y-axis in hertz. And what we can see here, this is pretty typical for human speech, starting at 0, of course, that's a good origin, 0 and 0, but it goes all the way up to about 7,000 hertz. And human speech, as we saw on the previous slide, has these formants, that the sound frequency that's generated in the vocal folds is shaped and twisted, modified, to have these harmonics that get emphasized. And it's the emphasization of those harmonics, and the peaks of which is what we call formants, that allows for the identification of a particular sound to be either a vocaled or a voiced consonant, or an actual vowel. So I is going to have relatively high formants. So there's a fundamental frequency that's generated. In this case, this fundamental frequency, the very bottom frequency, or the bottom formant, this is around 300 hertz. This is probably a man speaking. And then for this person who's speaking I, then the next formant is actually around 2500 hertz. So you can see there's a kind of a space here. Now, how do I know that that's what these formants are and where the strength is? That is because I'm looking at the darkness of the mark, and that indicates the amplitude of the sound that's being generated. So the loudest part of the vocalization of this individual, you can see, is down here, because this is particularly dark. You can see it's relatively dark right at the beginning and then kind of lightens up. But you can see that there are these bands here, and this is very typical of human speech, that you have these harmonic stacks. And that the position of these different formants is going to be specific to the phoneme that's being vocalized. So in this case, this is I. U is relatively low. U, I, and then A, somewhere in between. One thing we all know and probably take for granted is the fact that language is learned, because it just becomes so natural to us. But this is a very interesting topic and intensely studied. Infants, they can distinguish phoneme contrasts at a very early age. They can distinguish these phonemes from all languages starting from birth, all the way up until about 6 months of age. And you can do various tests to see whether the infant is distinguishing these different phonemes. It's amazing what actually can be done, even with these very little young infants. Now they start to get language-specific perception that we can identify between 6 and 10 months. And we also know that infants at 3 months old, they can produce vowel sounds. They start kind of babbling at around 7 months of age. And then they start to produce specific language at around 10 months of age, 10 to 12 months or so. And it's typically around a year old is when we start to see the first words, although there's a lot of variability when those first words appear. But there's also, importantly, there's a critical period that's necessary for language. So a child will need to be exposed to a language within the first 8 to 10 years in order to have true fluency, the ability to fully understand a language and to produce the phonemes like a native speaker. We can see this this critical period is most obvious in humans when we talk about the age of acquisition of a second language. So this is work done by Patricia Kuhl. She's done a huge amount of work on the development and neuroscience of language. She's a professor of neuroscience and language pathology at the University of Washington in Seattle. And she just gave a talk here at Virginia Tech in 2019. Very interesting talk. So, and she's done a ton of work on this, and this is one of her studies looking at language learning and at the age of acquisition of a second language, which is going to be indicative of and shaped by this closing of a critical period. So we know native language obviously starts very early. Almost every single infant is exposed to language at a very, very early age, and they have high language fluency. When they are exposed to a second language by the ages between 3 and 7 years old, they show language fluency that's basically the same as someone who was exposed to their native language or, say, a second language at infancy. A very high fluency as long as you learn that language within that critical period. And it's between the ages of 8 to older when we start to see declines in language scores. So if a child has been exposed to a second language between the ages of 8 to 10, 11 to 15, and 17 to 39, no longer a child there, we see language score goes down quite a bit. Ironically, in the United States, it's often the case that in most educational settings, we start learning second language in schools at the ages of 11 to 15, and consequently, we have a reputation of being a nation of people who can speak only one language, unless you were lucky enough to grow up in a household who speaks 2 languages. But if you ended up learning your language at high school, you probably didn't learn that language very well. And that's going to be very different than the kind of experiences that are seen in other countries, such as in Europe, where they get exposed to second and even third languages very, very early on, and their language fluency is much higher, which is one of the reasons why, for instance, English proficiency is incredibly high across Europe, because they start learning English between the ages of 3 and 7. So the process of learning language in humans is done through what you might characterize, or is characterized, as Motherese. So that's the way, we all are familiar with this, that's the way that adults will talk to infants. When we are talking to infants, we're using this Motherese, and it's characterized as using speech that is slower, exaggerated, and the vowel sounds are clearly articulated. So let's just give an example of adult-directed form. What you can see here, this is what's drawing here is the fundamental frequency, the F0. So this is the very first formant. And so we can just draw a line where that is. There would be a harmonic stack here if we could see the entire sonogram, but we're just drawing out the basic form. Then we can draw the bottom, basically. So you can see it's all in one line. This person is speaking with a single tone. It's around 350 hertz or so, 300 hertz. This might be female. Males, their voices tend to be around 200 hertz or a little bit less. So if I were to say this, it'd be, I had a little bit. And the doctor gave me Benedictine for it. Pretty monotone, pretty flat. And that's how you would talk to an adult. But if you're talking to a baby, you're going to give infant-directed speech, and you're going to use Motherese. You're going to use acoustic stretching to make the vowels contained in Motherese a lot more distinct, and that the stretching of the sounds is found in actually in all languages, which I'll explain in just a sec. So when we're doing infant-directed speech, it's going to be, can you say ah? See, it's going up. Ah. Say ah. Hey, you. Say hi. Hi. I can't do that one. So that's infant-directed speech. And as I said, this stretching of sounds is found in all languages. So this is in English. This is showing kind of the range of frequency stretching that happens at different vowels, I, U, and A. When for adult-directed speech, which is in blue, down here, adult-directed, and in red is the infant-directed speech. And you can see that this triangle of where these fundamental frequencies is found, the F1 versus the F2. So here's the F1 for you is around 350 hertz, and then the F2 is around 1300 hertz. That's for infants, but for adult-directed speech, it's actually a little bit higher, and you can just see that the range at which we can stretch our different frequencies is more narrow. The stretching of these vowels, when we direct it towards an infant, is much bigger. This is true for English. It is also true in Russian, which has some of these same phonemes, and Swedish also has similar phonemes, and you can see that Motherese is used in Swedish, Russian, English. Really any language that you're going to look at, you're going to find this kind of stretching. So why the heck would we do this? Well, it turns out infant-directed speech might assist infants in learning speech sounds. Mothers who stretch the vowels to a greater degree have infants who are better able to hear the subtle distinctions in speech. And these two factors are actually correlated. That's shown here in C. So this is showing the kind of stretching that occurs. So that's a measure of like the degree to which stretching occurs. And there's going to be variability in which this area. So they're basically calculating the area of this stretching that occurs for different mothers, and then looking at the performance of speech perception of the infant. And you can see that mothers who use a lot of large vowel stretching, they have infants that tend to perform better than those infants who are exposed to a smaller range of vowels. So this stretching, this Motherese, at the very least, seems to actually lead to better perception of speech and better performance in infants at a really young age. So it turns out that our ability to distinguish phonemes at a very early age, it appears that infants essentially have the fundamental features that are needed to learn these different phonemes, but we only really retain the phonemes that we actually end up hearing. So let me walk through some results that came out of Patricia Kuhl's lab on this process. So this is one experiment that she had done to demonstrate this. She was looking at infants from English-speaking American households and then also Japanese-speaking households. And she was testing them, these infants, at 6 to 8 months of age, seeing how well they could distinguish certain English phonemes. So a great example of this that's distinct to English-speaking households would be R and L. So for native English speakers, we can tell those things apart, but for a lot of Asian languages, such as Japanese, they actually have a hard time with even perceiving the difference between R and L. And certainly making those distinguishing between those two is tough for them as well. So these infants, they are tested at so that we test to see how well they can distinguish those different phonemes. And it turns out at 6 to 8 months of age, the American infants in red and the Japanese infants in blue, they both can distinguish the phonemes equally well. Not super great, but better than by chance. You can see it's around 65% correct when they're able to distinguish at 6 months of age. And then they test these same infants 10 to 12 months later. So during this time, the American infants are being exposed to more and more English, and they're learning more and more English, and they're getting a lot of exposure to R's and L's, whereas the Japanese infants are not. And so during that time, the American infants, they get better at distinguishing these different English phonemes. You can see they go up in their performance in distinguishing English phonemes, whereas the Japanese infants, they actually get worse. So a 10 to 12-month-old infant from a Japanese household actually ends up doing worse on distinguishing English phonemes than a 6 to 8-month-old Japanese infant in distinguishing these phonemes. And so this is an illustration that's just showing, and the idea is that as infants, we are blank slates, and we have the ability to perceive basically all of these different phonemes, and hopefully with the ability to produce them later on as we start to babble and produce words. But if we don't get that exposure during this very early window, we do not have that ability, and they're no longer retained. So when babies hear language, it can actually induce neural activity that can be recorded using an electroencephalograph cap. Look at this cute guy wearing this encephalograph cap. And this can detect event-related changes in potential. So when a baby is hearing its own native language, it actually has a very large mismatch in negativity. So there's a change in the overall potential that's perceived here, which is an indication that there's some sort of perception happening here. When this same infant is listening to a non-native language, there's a much smaller response. So the native language is inducing specific changes, and this is even before this baby can produce language, but it's having a perceptive difference that we can observe in its change in neural activity when it's hearing native language versus when it's hearing non-native language. And so these things are actually being manifested already in the brain. Turns out that personal interaction is actually really critical for the acquisition of language, and this includes second languages. Because it'd be great if we could just plop the kid in front of the TV and they can learn second language. Well, Patricia Kuhl had done an experiment to really test this hypothesis. She had children that were exposed, live exposure, to a Mandarin native speaker, or she had children that were getting the same kind of instruction from the exact same Mandarin native speaker, but through a television. And then so these were children that were approximately 10 months of age getting exposed to this. And these were American infants that did not come from Mandarin speaking households. They had 12 sessions of this with either the native speaker or with the television intervention. American infants that got the live language instruction actually learned to discriminate Mandarin phonemes much better than those that did not. And this is just 12 sessions that these children were getting. And you can see for the American infants that were exposed to Mandarin, their percent correct of distinguishing and identifying Chinese phonemes was much higher than American infants that were exposed just to English. For those infants that were exposed to television, and then they also had some other infants that they just played the audio to, no television at all, those children's abilities to distinguish the different phonemes was no different than children that got no instruction whatsoever. And you can see for the monolingually raised infants of the same age, those children that come from a Chinese household were somewhat similar, not significantly different from these American infants that got the 12 sessions of Mandarin instruction. So just 12 sessions of Mandarin instruction at the ages of around 10 to 12 months increases these children's ability to have to process and identify. Now that we've learned a little bit about how humans acquire language, now we're going to talk about some of the underlying neural pathways that are involved in processing language. So there's a lot of history for this. In fact, this is when you learn anything about the history of neuroscience, you're going to learn a lot about the guys that have identified these language areas. So we're going to learn about that a little bit. So one important region is called Broca's area. This is identified by a guy named Broca in 1864. This region is dominant. It's dominant on the left frontal lobe, located here, relatively ventral on the frontal lobe, and it's critical for the articulation of speech. We also have Wernicke's area, which is here. It's kind of at the intersection of the temporal lobe. It's sandwiched between the auditory cortex and the angular gyrus, and lesions of this disrupt normal speech. We obviously are utterly dependent upon motor and somatosensory cortex, motor cortex here, somatosensory cortex here. These things have to be happening and working in order for us to be able to produce language and control the muscles that of the vocal tract. Auditory cortex is used to perceive speech and allow us to learn the different phonemes so that we can produce normal speech as an adult. We also have visual cortex. Obviously, that's critical for the written portions of language, as well as perceiving hand gestures, facial expressions, which is also another important part of communication, and writing. So Paul Broca had identified this critical brain area that is now named after him, and identified that patients of his that had certain specific speech deficits had damage to particular parts of their brain. And this was eventually what we call Broca's area, this part of the frontal cortex. He had a few patients that, in particular, that are very famous cases in neuroscience. One guy, his last name was Leborn. He could only say the word tan. He couldn't say any other words. The only word he could say was tan. And he had this superficial lesion to the left posterior frontal lobe. There was another person, last name Lalong. This person could only repeat 5 words, and this person had a lesion in the same area. see kind of damage here. And so these brains are actually preserved. They're very famous brains. He examined other patients had similar speech deficits when they died, examined their brains, saw similar issues with lesions within these areas of the brain. He identified this area and referred to it now, this part of the frontal lobe is now called Broca's area. This is an example of someone with what's called Broca's aphasia. This patient, he experiences, and patients who have Broca's aphasia, they have difficulty speaking at all. They have what's called non-fluent aphasia. So their comprehension is generally intact, and they just have a lot of trouble finding the right word and putting the words together so that they can produce what is normal speech. And for these people, it's really a very frustrating exercise, because what will have happened for a lot of these people who have Broca's aphasia is that they've had some sort of stroke or maybe an injury that's damaged Broca's area or areas tied to Broca's area. And now they're having this issue with the inability to produce normal fluent speech. So they can't really find the word. And some of us will struggle with trying to find that word. But imagine if you had that issue for basically trying to find every word. And that's essentially what Broca's aphasia is. And it's really frustrating, because you know how frustrating it is when you can't find just one word. You're like, oh, I know that word. What is that word? What is it? Imagine doing that for every single word. And this issue of trouble of finding the right word is called anomia. Can't name the word anomia. And so I can't show the video for YouTube, but I'll put a link to the video right here. There's the link right here. It's also in the video description below, so you can look at this. And it's really interesting to see this person speak. Carl Wernicke is another famous neurologist. He described another type of speech and language deficit. These speech and language deficits are referred to as aphasia. And these cases that he was working on, these people had comprehension deficits. So he identified these individuals had a lesion at the junction of the temporal, parietal, and occipital lobes. We talked about before. This is called Wernicke's area. So in these cases, speech is actually quite fluent, but it doesn't actually make any sense. This kind of speech deficit is also called fluent aphasia, and it really kind of sounds like word salad. These people also tend to struggle with following commands. You explain something to them, and they can't really do it. That's because they have an inability to comprehend what is actually being said. It also seems like that these people are completely unaware of their errors. So they're producing the language, and they're having what seems like the kind of the start and stop of what you might be saying to fill in the spaces between to have a conversation, but the words are almost completely meaningless, and it really does sound like word salad. So I encourage you to look at this video as well. Again, there will also be a link in the video description below. A very interesting case. So yeah, aphasia, just to summarize this again, this is the loss of the ability to understand or express speech caused by brain damage. And this is a nice summary of the differences between Broca's aphasia and Wernicke's aphasia. Halting speech in Broca's aphasia, there's fluent speech in Wernicke's aphasia, and these people just have the ability to have comprehension, but they just are unable to create normal words in the proper order, have the proper syntax and grammar. Whereas Wernicke's aphasia, they seem to have all those things together, but they have very poor comprehension. So this is starting to show that these different areas may play a very important role in some of the fundamental features that are necessary for language comprehension and production. This is the comprehension portion, and this area clearly is very involved with the production side. So it's not simple, though, to say, hey, this is the production area and the Wernicke's area is just the comprehension area. we have learned an awful lot about the connections and the interactions of these different areas. And it turns out that the language and auditory centers are actually highly interconnected, and that there are reciprocal connections from many of these different language areas. So there are connections from Wernicke's area to premotor cortex. There's connections from Broca's area to Wernicke's area, auditory cortex to Broca's area. There's also interactions with motor cortex, obviously. So there isn't any sort of clear pattern. We can't just say that this is the comprehension portion right here, comprehension, and that this is the production side. It's probably an important dance and balance that happens between all these areas that it's critical for this. So this is an interesting diagram summarizing data from 117 patients that had language areas that were mapped using electrical recordings at the time of surgery. And the number that's within each of these circles indicates the percentage of patients who showed interference with language in response to stimulation. So when they're stimulating way down here on the temporal lobe, they saw no effect on language. But you can see for, in this case, way down here, 5 of the 117 patients had problems. When you get closer to Wernicke's area, you can see that does indeed seem to be like this is a major language center, but it's not universal. You can see that maybe about a third of all the patients had trouble with language processing here. And the same is true for Broca's area. In fact, right in the middle of what's traditionally considered to be Broca's area, only 18 patients out of 117 experienced problems. But right here is around 50, 36, 42. And then if you got into the motor cortex, you saw some pretty substantial effects, especially when you're down here in the head and face regions. 79 of 117 patients had problems with producing speech. So it's not clear that there's like any one particular pattern that's universal across all humans. There definitely appears to be this regional speciality, but there's going to be a lot of individual variability. And that's kind of the state of our knowledge of the location of these areas in humans at this point. There definitely appears to be regional specializations, but the interactions are actually quite complex, and it's definitely not universal across all human beings. So I mentioned that these areas are sort of focused on the left hemisphere, and that's because there appears to be some really important lateralization that happens with respect to language. So this is one of the first parts that we're going to be talking about, lateralization. One of the first and only parts. But there are many different kinds of behaviors that are lateralized, meaning it primarily seems to be driven by just one side of the brain and not so much by the other. Language is one of these classic systems that appears to be highly lateralized. So as we have learned, the brain is largely split, where the experiences that is perceived on one side of the body, such as visual input, is perceived by the other side of the brain. This is true for somatosensory perception and also motor control. We have this flip for a lot of the body, where one half is sensed by the other. But there's sharing that happens, and the sharing that happens between the hemispheres is most of this between the two hemispheres goes through what's called the corpus callosum. So this is a bundle of axons that goes not just through this portion on the coronal plane, but it's like a sheet that goes across the lateral ventricles and connects the different hemispheres. So there's about a lot of information that can pass from one side to the other. Now there are epilepsy patients. These are patients who have spontaneous seizures. Their seizures can be so bad that they need some sort of surgical intervention to help ameliorate some of the problems that they have. For these patients, what happens for these patients is that for some patients, they will have seizures that are so bad that starts on one side of the brain, and then they will spread via these axons through the corpus callosum to the other side of the brain. And one surgical intervention that can be useful in ameliorating this problem is just cutting the corpus callosum so that one side of the brain can no longer talk to the other side. These patients are known as split brain patients. They generally are pretty much okay, but it turns out that there's some really interesting things that they can't do, and that it reveals some important details about how the brain processes information. Speech is one of those things that we've learned a lot from split brain patients. So we're going to talk through some basic experiments that's been done with some of these split brain patients that shows that language is largely lateralized. So in a split brain patient that is put in front of an experimental setup like this, some objects in front of this individual: a red ball, green cube, a blue pyramid, and also a word here, said ball. So like the word appears, and then he's got to pick up the ball or something like that. That would be the case if he could perceive these things. So interestingly, if these things are on the left-hand side of this individual, so they're not in the right-hand side of their visual field, these people cannot describe the objects or read the words that are in their left-hand side of their visual field, but they can actually draw what they saw, which is crazy. So they can draw that they saw a cube, but they cannot articulate that they actually saw a cube. Why is this the case? So if you think about it, if it's on the left-hand side, that means it's largely being perceived by the right part, the right half of the brain. So let's walk through what this looks like here. So in a normal individual, you put a cube in the left part of the visual field, and they have to stare straight ahead. They can't cheat. They can't turn so that the cube is in the center or in the right part of the visual field. But, it's within the visual field, so they should be able to see it. This information from the nasal portion of the left eye, the temporal portion of the right eye, all of it ends up on the right-hand side of the brain. And then what would normally happen is that this information would make its way through the corpus callosum to the other side of the brain, and then eventually make its way to some of these critical language centers, such as Broca's area. This information, because Broca's area is on the left-hand side, and information and objects and things that are on the left-hand visual field would have to make their way over here to the right and then be passed through the corpus callosum. For those individuals that are split brain individuals, the light information makes its way to the right visual cortex, but it can't get to the other side into Broca's area. So they cannot perceive the objects. They perceive them, they can see them, but they cannot describe the objects because it doesn't make it to these language centers. When you take that object and then now you put it into the right visual field, now the person is perceiving it with the right side of the left side of their brain. So you can see that it's hitting these portions of the retina, and that now the information is getting to the left visual cortex, and it's being passed forward to these what pathways in the temporal lobe, allowing the left side of the brain, where Broca's area is located, to and Wernicke's area, to perceive these things and then process our language perception of what this is and say this is a yellow cube. Pretty cool. So we talk about language being the left hemisphere sort of function, but it turns out that there's lateralization related to language and our other important salience features about language that's also controlled by the right-hand side. It's not like the right-hand side has nothing to do with language, it turns out. So there's a wide range of hemispheric functions that can be evaluated using this method. Even in normal subjects, we can do a variety of things. So this list right here is showing some of the different functional abilities for the left and the right hemispheres that we've learned from various split brain patients. So in the left hemisphere functions, again, it's obviously analysis of the right visual field. We have stereognosis, so perceiving things with our right hand. Lexical and syntactic language is processed in the left hemisphere. So that's where Broca's area is involved. And we also have writing and speech as being critical things that are regulated by the left hemisphere in most patients. The right hemisphere, however, does important things too. It actually is involved with the emotional coloring of language, such as prosody, which we refer more specifically is referred to as innotation and stress. So prosody is, sort of like, geez, can't you do this right? So putting some innotation and stress on certain things, and you can tell that I'm sort of emotionally involved with whatever I'm trying to communicate. The right hemisphere damage can also lead to a condition called aprosodia, which is where these individuals will have a hard time providing prosody to their language. They have a hard time giving innotation and stress and not stress like, oh, I'm stressed out, but stressing and coloring the language so that it carries emotional salience. Turns out sign language is also lateralized as well. So for these individuals, they don't speak at all, but for these individuals that have learned American Sign Language, it clearly has components of language where there is a series of syntax and grammar and like an order that the words have to be put into place. A lot of these things are left hemispheric dominant, and damage to the left side in sign language users leads to sign language aphasia. So people who have damage to Broca's area end up using the wrong signs, or they have a difficult time finding signs. And then people who have damage to Wernicke's area, they appear to actually they can sign normally, they can make the signs, but their signs are completely incomprehensible and they're signing incorrectly. Totally crazy. So they're doing the exact same thing, but instead of it being involved processing the muscles of the vocal tract, instead, for those individuals that have to use sign language to communicate, they still end up with the same problems, but it's because they're using their hands to process language. Some of this lateralization can be observed even anatomically. There's a portion of the temporal lobe that's larger on the left-hand side than it is on the right-hand side in nearly all individuals, more than 90% of individuals. This part of the temporal lobe is called the planum temporale, and it's kind of towards the top of the temporal lobe, and it's right at kind of at this juncture that's really critical for auditory perception and language processing. So it's likely that this is going to be some of the major reasons why we see this lateralization in language processing. And we can even see this anatomical difference when we look inside a brain. So if we were to slice a brain like this and do a transverse section such as this, you can see that this portion of the temporal lobe is larger than this portion here. This is pretty typical. In fact, it appears to be something that starts even at birth. So infants have a planum temporale it's at least 50% larger on the left than it is on the right. And this magnitude of difference is maintained even as brains grow as we get to our adult brain size. And see here, what we're looking at is an MRI image where we can see on the left side here and the right-hand side here, some of the folds that are different. And in fact, this anatomical mismatch or this lateralization is one of the largest lateralizations that we can see in the human brain. This part of the brain is very distinct on the left-hand side than it is on the right in most brains. We can observe changes in brain activity using a variety of methods. We've learned about some of them, such as calcium imaging, fMRI, or doing in vivo electrophysiological recordings. Another method is using positron emission tomography, or PET. This uses radioactivity in order to do this. You have radioactive chemicals to measure changes in brain activity. A subject is injected with a radioactive chemical such as glucose. So this glucose is loaded with carbons. The chemical that is often that's used to make the radioactivity is radioactive carbon. As the carbon decays, it creates this positron, which is the opposite of an electron. It's like a positive electron. And it interacts with an electron. So it'll shoot out and interact with an electron to create gamma rays. These gamma rays then can be detected by the detector. So this is a PET scanner. You can put a person in here, inject them with a bunch of radioactive glucose, and then wherever the brain is using a lot more glucose, then you're going to see more radioactivity in those areas, and this detector can detect the light coming from that particular part of the brain. And so this is what a PET image looks like. Scientists have done a ton of experiments to understand how parts of the brain that are involved with language and how they function depending upon doing different kinds of language tasks. So individuals in a PET scanner that are instructed to follow a special display. The left panel here, this indicates the task that's being presented to the person prior to scanning, and then the PET scan is shown at the right. So these language tasks, for instance, you have to listen to words that are generating word associations and elicit activity in Broca's and Wernicke's areas, such as expected. But we also see that there's activity in primary and association sensory and motor areas for both active and passive language tasks. So some of these passive tasks is such as listening to words, and we'll see activity in some of these areas. Speaking words, obviously we see activity in the motor cortex, but we see activity in some of these areas. And this indicates that the language processing involves many cortical regions in addition to the classical language areas. It's not just Broca's area and Wernicke's area. That in fact, depending upon the task, you're going to see a constellation of brain areas that get activated. There are different regions in the temporal lobe that are activated by different word categories that we can see using PET imaging. So depending upon the kind of category that we're talking about, get different areas that get activated. So again, this is some more PET imaging. The dotted lines, they show the locations of relevant temporal regions in these horizontal views. You should see that the different patterns of activity in the temporal lobe in response to each stimulus category. So we got 3 categories that we're illustrating here: people, words, people-related words, animals, and then tools. And you can see that the pattern isn't the same across all of them. So people has a very strong activation on the left hemisphere towards the more frontal portion. But then not a whole lot of activity down here, whereas animals has activity in this area as well, but also some activity at the more medial portions of this area. And then tools, it appears to also be activating this portion here. This is likely to be Broca's area. But then we have another area that appears to be a little bit more active, a little bit more lateral, probably someplace else in the temporal lobe, a different categorization portion that you might think of as the tools area for this particular individual. So you're going to find different categories of words activating different parts of the brain. So I wanted to talk about some additional wrinkles to our understanding of language. One interesting feature that we've learned about language is related to sensory substitution. So sensory substitution is when the loss of a sensory modality leads to the changes in remaining modalities. For some individuals, they are born blind, and we know that some of these individuals may have enhancements in their auditory processing or in their touch processing. And so we're going to learn just a little bit about that and some interesting experiments related to that. For blinded individuals, so these are individuals that are born blind, and they're taught how to read braille. So they've been reading braille for a long time. When we do fMRI scanning for these individuals, when they are reading braille, we see activity in the occipital lobe in these individuals. And if you remember from the visual portions of this class, those are the visual centers of the brain. This is going to be V1, V2, V3, V4. That's what you're going to mostly find in the occipital lobe. These individuals are blind. They are not able to see. Yet when they're reading with braille, they're using their visual cortex. So it's almost as if they are reading, using the visual centers of their brain with their fingers. So these visual areas have now been reorganized and are now being utilized in the these blind individuals, these congenitally blind individuals, to perceive braille. Very interesting. You can test the critical role for different brain areas for doing different functions by manipulating brain activity using what's called transcranial magnetic stimulation, or TMS. This can be used to block activity in specific brain areas. So I'm going to describe for you an experiment that's been done to test this. So this is what a TMS machine looks like. So there's the magnetic field that gets passed within this device that can shut down parts of our cortex, cause certain kinds of changes. Pretty crazy, actually. And in this study, they did this experiment to individuals who could see, but knew how to read braille, and then individuals who were blind, and those individuals obviously could know how to read braille too. And they shut down different parts of the cortex. They did a midfrontal portion. They hit the somatosensory and motor areas. They hit parietal areas, and then they also hit occipital areas, more visual-like areas. And they did this while they were doing these tasks to see how well the individuals did the tasks. For those individuals that were sighted, so let's start over here. These are the sighted individuals. When they knocked down the somatosensory and motor areas on the contralateral side of their brain. So if this individual was reading with their right hand, they were knocking down the left contralateral somatosensory and cortical area. This led to a much greater error rate. That makes sense. So if you're touching with your fingers on your right hand, and if they knock down the somatosensory cortex so that they're disrupting activity in the somatosensory and motor cortex on the left-hand side of your brain, you're going to have a harder time reading braille. So you're going to get more errors. For blinded individuals, the somatosensory and motor cortex did lead to some error rate that was greater than some of the other areas, but it really didn't differ that much. They found for the area that was most effective in knocking down the ability for blinded individuals to read braille was the mid-occipital, where the visual cortex is located. So by blocking visual cortex function, blinded individuals were no longer able to read braille. Remember, they cannot see, but they're using their visual cortex to read braille. Pretty cool. So what we've learned from modern imaging, from PET scans and fMRI, is that multiple brain areas are critical for language, not just Broca's and Wernicke's area. We've learned that brain imaging and stimulation studies have revealed that widespread brain areas are involved in both hemispheres in language. It's not just on the left. There's also prosody and other emotional salience that's added on the right-hand side of the brain. We have language processing skills that involves a collection of interacting subsystems for different language skills. And there are many different components to language processing. We have understanding words and sounds, the meaning of words, grammar, naming objects, producing speech, etc. So there's a lot of different things that are needed in order to make language. There appears to be multiple areas of the brain involved with these different processes, and they appear to be malleable in instances of sensory substitution. Okay, so we talked a lot about speech and language. Now we're going to talk about a different kind of vocalization.
Time seeing this.
It's also a critical component of communication, and that's called laughter. So
actually, that's not so bad.
Going to hopefully these videos.
And there you go. There's
of course
the Steve Butler mask with Steve Butler.
You'll have to do the PowerPoint to look at these videos. I was hoping that we could have a little bit of laugh. This guy is going to put on a mask, and it's a mask.
Okay.
Face, and he just starts laughing. It's super hilarious. And then this is
all right.
This is probably like some guys that looks like they might be in a dorm, and
I can't take myself seriously while I'm wearing this. Okay.
Chair. He's got a harmonica in his mouth, and they hit him in the shin, and then he starts making these noises with the harmonica, and they just start cracking up, and it's pretty hilarious. Yeah. So laughter is a universal phenomenon seen in all human cultures. It's obviously that we use it to express joy, and it's when you think about laughter, it's really kind of a weird thing. We take it for granted, But all of a sudden, here you are experiencing joy, and you're going to start creating these weird noises. And then you're going to start maybe encouraging the other people around you to also be making those same weird noises. And so it's clearly kind of a social event. And in fact, we know that this is the case. In fact, this is part of the reason why a lot of sitcoms on TV have had to add laugh tracks. So you add laugh tracks because if you're sitting there in front of your TV and you're sitting there alone and something funny happens, the writers have this intention of wanting to share this joyful event with you. So do the actors. But, they're acting and they're doing a part. So in order to encourage you to laugh, they have learned that if they play a laugh track, that is going to get you in the mood to be laughing as well. And so that you can share with it, even if you're alone. They're manipulating you to create that social bond. So laugh also, it comes in many forms. We have polite laughter, uncomfortable laughter, joy. You can think of many others. And human laugh is a series of short 75 millisecond notes during exhalation with intervals of about 200 milliseconds and with a pause in normal breathing. So you basically stop normal breathing. So it's really bizarre that we would do this thing that would create all this noise and also not even breathe. And you would think that this might make you vulnerable to, say, predators or that kind of thing if you're thinking about like cavemen and whatnot. But it's something that all humans do. And it's because it's a critical component of social bonding and play. When you manipulate these vocalizations and you start changing them around, laugh then becomes unrecognizable. Then it just sounds like weird noises. And so we know what laughter is, and in fact, we know what fake laughter is. So when people try to make a laugh and not give a genuine laugh, we can distinguish between those two things. So we're really good at telling laughter. Laughter is seen in other animals as well. This is a cute video of bonobo chimps that are getting tickled and laughing. So I encourage you to take a look at this video. There'll be a link description below. When they get tickled, they laugh. It's very cute. Believe it or not, even rats can laugh. In fact, there's a lab in the School of Neuroscience, Mike Bowers' lab. He has an entire course on the neuroscience of language and is really an expert in understanding language. And he studies the vocalizations of rats. And he's been categorizing these things and determining the kinds of contexts in which the vocalizations of rats is used. And one thing that he has talked about, and it's really pretty hilarious, is that rats will giggle when you tickle them. And so there's been some experiments to show exactly how this happens. This is some images from a paper that was published in Science about 4 years ago. This is a scientist tickling a rat on the belly, the ventral side. And they make these noises. They make these high modulated trills and combined. They have modulated sounds that they make. They make these trills, and then they do combinations of these things. This is at really high frequencies. As I told you before, human speech tends to be around 300, 400, 500 hertz. This is 50,000 hertz. This is kilohertz, the scale. So 50,000 hertz. This is well above the perception of human ears. So we can't even hear it. But what we do know is that when you're in a laboratory setting, or even with like a pet rat, that if you start to tickle it and start to play with it, rats are actually really social animals and they love it. So they will chase you around and they'll want to be tickled. So you move your hand away from the rat, and so you may have tickled a little bit. It thinks that it's now time to get tickled. You move your hand away, the rat will come up to your hand and want to be tickled. And it'll start making these noises that sound like giggles. So these ultrasonic vocalizations that we're calling giggling, so that's the USV rate here. So how many times they're doing this per second. So prior to the tickling, there's really not a whole lot, but then during the breaks, there appears to be some giggling that occurs. If you just touch the tail, it's a little bit more than obviously before or the break, but they really start to giggle a whole lot when you do dorsal tickling, so tickling their back, or tickling their belly. Belly is the most. You can see that that's even higher than tickling the back. And when you put an electrode down into the somatosensory cortex, into a portion of the cortex, and you tickle them, you can see activity. So they're looking at changes in activity in this portion of the somatosensory cortex. During tickling, there's an increase in the rate of firing that happens for this part of the cortex. And then when they stop, they take a break, the firing rate goes down. And then when they start tickling again, that's these dark areas here, it goes up again. When you do just gentle touch on the dorsal side or the ventral side, you do get an increase, but it's not as much as if you were tickling them. When you do tail tickling, it's higher. And then this is just them chasing the hand. So they're not even being touched at this point. They're just running around chasing the hand because they want to get tickled. And there's an increase during this point here. So this appears to be a neural correlate of giggling or laughter in rats. Even crazier is that in these rats, they lowered this electrode into this area of the brain. And when they stimulated layer 5 of this same part of the somatosensory cortex, it elicits giggling behavior in rats. So that's what we're looking at here. So this is a series of stimulations. And then this is showing a change in the ultrasonic vocalization rate. So when they start to stimulate in this area, which we know is part of the somatosensory cortex and seems to be touch sensitive, when they stimulate in these areas, they get an increase in the USV rate. It's relatively short. You can see it lasts less than a second, but it's a huge increase relative to baseline. And so that you can elicit very short, quick little giggles as if they're being tickled by stimulating these areas. So clearly, this kind of vocalization is, obviously universal to humans, but it even seems to be shared in rats as well. So to wrap this lecture up, here are the key questions. Again, you should really go through these key questions. What is the definition of language? What are phonemes? What are the physical mechanisms that the body uses to create speech? What, that vocal tract, the vocal folds. What are the important features of language acquisition? And what is the critical period for language? You should know about Broca and Wernicke's contributions to neuroscience and what is aphasia. What is the role of the left and right hemispheres with respect to language, prosody versus comprehension and production? What is the modern imaging techniques shown about brain processing? And what parts of the brain are used by blinded individuals to read Braille? So that's it for this lecture, and we'll see you next time. Bye-bye.